Okay, hello, welcome back to the Science Shack for what I hope will be quite a nice quick one today. So we're going to split meiosis up into two parts, the basic stages and a bit about life cycles and why it's important, and then the second stage is going to be where this variation, which of course is the point of this topic, actually comes in. How do we end up with variation? Okay, so let's crack straight on into it. This is the bit on meiosis, right, the idea of cell division. You already looked at mutations, genetic variation in that way, so the idea of changes of chromosomes, changes of DNA, and to generate different alleles. Now we're going to look at how we actually split those alleles up to make new combinations that have never actually happened before, and when in a life cycle that takes place. Now there's a huge box there that says students should be able to, which basically means you should be able to do an awful lot of those things, obviously, right? So there's a lot of hints and tips as to what the exam questions are going to be. So if we move on, I'm assuming some basic prior knowledge. I'm assuming that you understand chromosomal structure, cell structure, when something is a chromosome, when it's two chromatids, what I mean by a pair of chromosomes, right? See previous rant about just an X-shaped object is not a pair of chromosomes. A pair of chromosomes comes from your mum and your dad, right? That X-shaped thing is one chromosome that's been through DNA replication. Very important that we get the distinction there. Okay, I'm also expecting you to remember mitosis with a T, right? I need you to know your prophases from your anaphases, from your metaphases, and so on and so forth. And then obviously basic genetic vocabulary, haploid, diploid, all that kind of stuff. Okay, I'm expecting those things as well. So, moving on very quickly. Let's look at a really simple life cycle here. Right, let's look at this. Oh, excuse me. Right, let's look at a very simple life cycle. We've got this. What happens in a basic reproductive cycle. So to start with, in your life, when you grow from your embryo, you are what we call 2N. You are a diploid organism. You've got one set of chromosomes from your mum and one set from your dad. Many divisions of mitosis with a T later. You are an adult. Okay. Then you start manufacturing your mature gametes, okay? Happens at different stages in male and female, which obviously generate 1N or haploid cells, the sperm or the egg. When they combine, they restore that chromosome number up to 2N or the diploid number. You then get an embryo and you get an adult forming there. So of course we need to basically be aware that in this stage here, right? So this stage here is where we've got meiosis, right? And the rest of those stages are mitosis with a T. And spotting when meiosis is occurring, when mitosis is occurring, is a standard exam question skill. So for example, if we move straight on quickly, if we move on quickly to this, right? This is a very simple life cycle diagram in humans, okay? The idea that on one side you've got diploid cells and on the other side you've got haploid cells. So from diploid to haploid is meiosis, and from haploid to diploid is fertilization, and then basically any time a cell just replicates, be it haploid or diploid, that is mitosis with a T. Okay, so this is our life cycle here, labelled on. It's a very simple one, it includes humans, but you might get given a more complicated one than this in an exam situation. So I'll give you an example that comes up quite often, which is generation alternation in things like mosses and ferns. Now they behave a little bit differently. So if we start down here with our diploid organism, so this is an adult diploid organism that we call a sporophyte. Okay, now our sporophyte goes through meiosis and generates haploid cells, just like us generating gametes. But the difference is those spores don't need to combine to grow into a new organism. Those spores can be sent out into the world and they can grow into their own organism, right, on their own. Okay, that is a haploid organism called a gametophyte. Okay, and that is all done by mitosis. Doesn't matter that these are haploid cells, if they're just replicating themselves over and over again, maintaining the same chromosome number, that is mitosis with a T. Mitosis of haploid cells, okay? So that is a new concept, right? That gametophyte there, then when it gets to maturity, just cells break off that, combine with cells from another organism, another gametophyte, restore the diploid number, you get a zygote in the normal way, that then goes through mitosis again and grows into an adult sporophyte. So every other generation, your mosses and ferns are haploid, diploid, haploid, diploid, haploid, diploid, okay? That's why we call this generation, this alternation of generations. And a really common exam question could be to identify where meiosis and mitosis are taking place in an unusual life cycle. So let's have a lock at a couple of those now. So what I want you to do is pause the video at this point here. This is an organism. Please read the bit at the side there. It's an organism whose diploid cell contains 14 chromosomes. So pause the video here. If it's a blue box, it's after a number. If it's a red box, it's a process, mitosis or meiosis. So correct numbers in the blue boxes, please, and correct words, mitosis or meiosis, in the red boxes. Pause video now. Okay, good stuff. Right, so hopefully you got the idea that we should have 7 in a haploid cell, 1 in, and 14 in the diploid cell there, okay, because the diploid number is 14, and then basically anything on one side over there is going to be 7, okay, and on the other side is going to be 14. So the idea is that down here we've got our spores that are formed. Okay, so spores are on the haploid side, so they're all 7. They're going through mitosis in terms of forming from haploid cells, and then only when they recombine at the top there, fertilization, do we restore that 14 number, and then everything on the other side is 14. So then it should be quite straightforward to work out that the one at the bottom there is meiosis, okay, because we're going from 14 down to 7. The rest of it, because we're maintaining that chromosome number, is mitosis. Hopefully that's clear. So I'll show an exam question for this, right? This is basically where we're asking the question. This shows the life cycle of a moss, right? So the idea that you've got the only parts of the spore capsule, only the stalk and the spore capsule are diploid. All the cells in the other stages of the life cycle of the moss are haploid. And the question goes, which letter A, B, C or D in figure 1 shows where meiosis occurs in the life cycle of the moss? Write the appropriate letter in the box provided. Okay, so have a look at that. Where is meiosis taking place? And of course, the answer is D there, the formation of those spores. All right, so moving on. So we're now going to go into meiosis proper, and I'm going to avoid the bits about where variation is built in, because that comes in the next video, but I want you to know the words, I want you to know the names by the end of this lesson. The idea that if you know your interphase, prophase, metaphase, anaphase, telophase, cytokinesis, it's not too difficult to learn meiosis, because all meiosis is two sets of cell division. Okay, it's 2 sets of cell division, so we just put a number after them. So meiosis has the same stages, right, but it just does it twice. Okay, so we've got interphase 1, prophase 1, metaphase 1, anaphase 1, telophase 1. We then get very short growth cycle with no replication of the DNA, because remember we're trying to cut the amount of DNA, cut the number of chromosomes in half by the end of this, okay? And then we go straight into interphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so this is meiosis 1, and this is actually where all of the variety gets introduced, all the variation, through 2 processes that I'm going to miss out for now, come back to in the next video. So they happen sort of up here in sort of prophase, metaphase stages. Okay, and if we go to this point here, meiosis 2, right, as you can see, we've already gone down to 1N, right? So we've already made our cells haploid, even though they've still got the same amount of DNA, they've only got half the number of chromosomes. Then we go prophase 2, metaphase 2, anaphase 2, telophase 2, and that gives us our 1N gametes that have half the amount of DNA as well. Okay, so it's important that you know the words interphase, prophase, metaphase, anaphase, telophase and cytokinesis from your mitosis lessons. The only difference for meiosis is we put the numbers 1 or 2 after them. Okay, so let's line them up next to each other, mitosis and meiosis. So as you can see, there's 2 cell divisions in meiosis and only 1 in mitosis. We end up with 4 haploid cells as opposed to 2 diploid cells. Okay, and the subject of the next lesson is the variation, the differences. So whereas in mitosis our resulting cells are going to be genetically identical, chromosomally, they're going to be clones, these 4 down here are all different. Okay, so these 4 gametes, these 4 haploid cells are varied. And those 2 points where variation come in are here, right? Independent segregation and a process called crossing over. And that is where the variation that makes every sexually reproducing organism unique from every other comes in. Okay, and that is the subject of the next video. Thanks very much. I'll see you then.